All right, so today we're going to talk about muscle. So the last section here is on muscle. And there are three types of muscle tissue. There's skeletal muscle, smooth muscle, and cardiac muscle. And they differ in a number of ways: in structure, location, function, and means of activation. When most people think of muscle, they think of skeletal muscle, okay? Which is how we move, the contraction of skeletal muscle. Skeletal muscle is under voluntary control, okay? In other words, if I want to lift up my arm, I just have to think about it. And muscles contract, and they result in movement of bones. Okay? So we say that skeletal muscle is under volitional control. Right? Whereas smooth muscle and cardiac muscle are not under voluntary control. Okay? They're innervated by the autonomic nervous system. Cardiac muscle is only found in the heart, and of course it's responsible for the heartbeat. Smooth muscle is also called visceral muscle. Why? Because it lines the walls of organs. That's what a viscera is, an organ. So it's found in the walls of hollow, visceral organs, like the stomach, small intestine, the bladder, the uterus. That type of hollow organ is typically lined with smooth muscle. Okay? Of all these types of muscle, only skeletal is under voluntary control. Now, when we talk about a nerve fiber, the term fiber refers to the axon of that neuron. When we talk about a muscle fiber, the term muscle fiber refers to the entire cell. Not just a part of it, the entire cell. Let's matriculate forward. I've drawn some pictures here of different types of muscle so that we can emphasize the differences. Okay? The first type I've drawn here is skeletal muscle. Notice that the skeletal muscle fibers or cells are cylindrical in shape. You know what cylindrical means?
Like a cylinder.
It's shaped like a cylinder. So the ends aren't tapered. They're not tapered. Skeletal muscle is striated. What does that mean, striated? It means that it has alternating dark and light bands. Another feature of skeletal muscle is that it has multiple nuclei per cell. It's different from most cells. And secondarily, the nuclei are peripherally located just beneath the cell membrane. And then finally, skeletal muscle, or as they say in Britain, skeletal muscle, it's under voluntary control, as I demonstrated earlier. Yes. Very good. Very good. Now, let's compare that to smooth muscle. Okay. We said smooth muscle doesn't connect to bone. A smooth muscle lines the walls of hollow organs. The shape is different. Instead of being cylindrical, the ends are tapered. So we say the cells are fusiform in shape. Fusiform means that simply that the ends are tapered like this. Okay? So the shape is fusiform. They have a single nucleus that's central in location. So whereas skeletal muscle has multiple peripherally located nuclei, smooth muscle has a single centrally located nucleus. When we talked about skeletal muscle, we said that it's striated. Smooth muscle is not. It's unstriated. We said that skeletal muscle was under voluntary control. Smooth muscle is not. All right. Now, let's compare that to a third type of muscle, cardiac muscle. The one thing you should notice here when looking at this drawing of cardiac muscle is that the cells are branched. It consists of branching fibers. Okay. Now, typically each cell has a single centrally located nucleus. So for instance, the nucleus is in the center of the cell. Okay. Occasionally, you're going to see two nuclei, but most cells have a single nucleus. Like skeletal muscle, it's striated, alternating dark and light bands. Unlike skeletal muscle, it's not under voluntary control. Unlike skeletal muscle, it's not attached to bones. It's only found within the heart. Now, a unique feature of cardiac muscle is the presence of what are called intercalated discs. Intercalated disc. Intercalated disc refers to the appearance of the cell membrane when two neighboring cells meet. You see this infolding? The cell membrane here on either side of the junction. It's kind of like corrugated cardboard, how it meets like this. Okay? It forms a thickening at the junction between these two cells, and that thickening is evident when you look through the microscope at the tissue. We call it an intercalated disc. Now, embedded within this intercalated disc, cardiac muscle, you have these little channels here. These are called gap junctions. You cannot see gap junctions under a light microscope, but you can see it if you had an electron microscope. And these gap junctions allow ions, charged particles, that are entering the first cell here as a result of an action potential. It allows them to spread from one cell to the next. So gap junctions allow the movement of ions and the passing of an action potential from one cell to the neighboring cell. So for instance, as sodium, a positively charged ion, enters this cell with an action potential, it can then enter into the neighboring cell and trigger opening of voltage-gated channels so that that action potential then spreads from one cell to the next. Okay? So it's exciting here that we have different types of muscle cells.
Yay!
Yeah. Now, here's a picture. Eventually, you'll have to be able to look through a microscope and differentiate the three types. Now, this slide is the intercalated disc is so prominent that it's not difficult to distinguish this as cardiac muscle. You see the striations. You see the centrally located nuclei. Okay. You see these intercalated discs. And you see some branching here of the fibers. Here's a branch here. So this is obviously cardiac muscle. Whereas in skeletal muscle, the striations are very prominent. Okay. The cells are cylindrical, so they don't taper. You have multiple peripherally located nuclei. So this is clearly skeletal muscle. And then smooth muscle, the fibers tend to be thinner, tapered, and they tend to have an interwoven appearance like this. So this is, but you don't see any striations, so this is smooth muscle. All right, so let's talk about some characteristics of muscle tissue that make it special. It's excitable or irritable. That means that it can receive a stimulus and contract. Okay? It can respond to a stimulus. And that response, that's number two, is contractility, the ability to shorten with force. Now, muscle is also extensible. You can stretch it. Okay? And on top of that, it exhibits elasticity, meaning that once you let go, it recoils, assuming the original length. So those are all some characteristics, all characteristics of muscle tissue. Now, let's talk about functions. We said earlier that skeletal muscle is responsible for all movement or locomotion. Watch this. Do the locomotion. Yeah, that is a song. Do the locomotion. Come on, baby. Do the locomotion. Come on, baby. Do the locomotion with me. All right, so yeah, that's a good song. Okay, so locomotion, and there was a dance that went with, like a line dance. Yeah. Like women love line dances. Whereas what you find is that men hate line dances. All right?
Nuh-uh.
In general. Now, cardiac muscle does this. Yeah, responsible for pumping blood throughout the heart. When the chambers of the heart contract, blood is pumped out into blood vessels. Smooth muscle contracts in a wave-like fashion. So that the smooth muscle surrounding, for instance, part of the colon. Let's draw the colon in here. Okay. The wall here has smooth muscle. It's going to contract in a wave-like fashion, and as it does so, it's going to propel the contents of the colon forward. Okay. Through the artery. That wave-like contraction within smooth muscle, what's the term we use for that? Does anyone know? The wave-like contraction? Peristalsis, right? Peristalsis. You probably learned that in high school, huh? Yeah. And then, what's that?
So I heard about caramel base.
Is that where you went, Mark?
Was I'm under open bell. True bell.
Yeah.
I always get, I don't know why.
Wait, what, you AOL?
Yeah.
Okay. Now, muscles also maintain posture and stabilize joints and generate heat. For instance, if we're too cold, what happens to our muscles? They begin to do what?
Stiffen.
They shiver, right? And in shivering, you're generating heat. If you're too cold, that's how you bring back your body temperature.
And it's harder to move.
Now, each muscle, and here we're talking about like the biceps muscle, or the triceps, or the quadriceps. Each muscle is a discrete organ. Remember that organs are formed of tissues. So it's composed of muscle tissue, but it's got to have more than that. It's going to contain nervous tissue, nerve fibers to provide innervation to that muscle, to trigger contraction. It's going to have connective tissue. Of course, you're going to find blood vessels because it needs nutrients and oxygen. Now, within muscle, there are three types of connective tissue sheaths. And these are called the endomysium, the perimysium, and the epimysium. Let's look at this muscle. Let's say this is the biceps muscle here. The individual muscle fibers are grouped into little fascicles. That's what we're seeing here, muscle fascicles, each of which contains individual cells, which could be called cells or muscle fibers. Now, the connective tissue sheath that surrounds an individual muscle cell, like this one, in white here, this connective tissue sheath is referred to as an endomysium. Now, the connective tissue sheath that wraps around the entire fascicle, we'd call that a perimysium. And then this connective tissue sheath that wraps around the entire muscle would be referred to as the epimysium. Notice that when the epimysium extends beyond the margin of the muscle here, it becomes the tendon, which attaches to bone.
So is the tendon like layers of the epimysium, or is it just?
The epimysium becomes the tendon.
Okay.
The tendon, remember, is dense regular connective tissue that connects muscle to bone. Okay. When it covers the muscle, it's still dense regular connective tissue, but its name is now the epimysium. So let's go back. The endomysium is the fine sheath of connective tissue. It's made of reticular fibers surrounding each fiber. Perimysium, fibers, connective tissue that surrounds a group of muscle fibers called fascicles. And epimysium is dense regular connective tissue that surrounds the entire muscle. And again, beyond the muscle, it becomes the tendon. Now, like any other organ, it needs a blood supply that can deliver oxygen and nutrients. Each muscle is going to be served by one nerve, an artery, which delivers oxygen and nutrients, and it's going to be served by one or more veins that are going to drain blood from that muscle. Okay? Now, that nerve that's serving the muscle is going to branch. The nerve is going to carry multiple axons. Let's go back. The nerve that serves that muscle is carrying lots of axons that are going to provide innervation. And in fact, each axon is going to branch. Okay? So each fiber, each cell is supplied by a nerve ending that controls contraction. Contracting fibers, in other words, active muscle, requires a continuous delivery of oxygen and nutrients that are delivered via arteries. And the waste have to be picked up and removed via veins. Yeah, contracting muscle requires oxygen. Oxygen is used to produce ATP. That's the form of energy needed for muscle contraction. Now, most skeletal muscles span at least one joint. Some muscles span more than one joint. Okay. And they're going to connect to the bones on either side of that joint. Okay. For example, let's take a look at skinning. Okay. This is the humerus. Do you remember that? This is the humerus. And let's say that this right here is going to be what? You remember what this is called? Ulnar, right. So there's going to be a muscle called the brachialis that's going to span this elbow joint. On one side, it's going to attach to the humerus. On the other side, it's going to attach to the ulnar. Now, when this muscle contracts, it's going to move the ulnar closer to the humerus, like this. In other words, it's going to cause flexion at the elbow. Okay. The attachment to the movable bone, okay, to the bone that moves, the attachment here to the ulnar is called the muscle's insertion. Okay. Okay. So the insertion is going to cause the ulnar to move closer to the immovable bone here at the humerus. The attachment here to the humerus, the bone that doesn't move, is called the origin. The origin. Now, some muscles attach to bone directly. That means there's no intervening tendon that extends beyond the muscle and then attaches to the bone. That instead, the muscle attaches directly to the bone. And I'll give you an example of that type of muscle. Look at these intercostal muscles. You see muscle attaching directly to the rib above and to the rib below. That's referred to as a direct attachment because there's no intervening tendon.
Because it doesn't need to be one, right?
No, it doesn't need to be. Whereas if you look at the biceps muscle here, okay, biceps meaning there's two bellies here. It's a bipartite muscle. So it's going to have two tendons up here, but it attaches via a tendon here to its origin. And it's attaching to the insertion here. It's a tendon. See that little white area? So that would be referred to as an indirect attachment, where connective tissue wrappings extending beyond the muscle serve as a tendon, or in some cases an aponeurosis. But it's connective tissue attached to the muscle that actually inserts or originates. Here, look, we show the brachialis again. That's crossing the elbow joint, okay, and produces flexion of the forearm at the elbow. What type of attachment is this? Is that direct or indirect? Indirect. What bone is the origin? The humerus. What's the insertion? Ulnar. Right. Now, this situation just tickles me here, because here we're demonstrating the biceps brachii. And what's nice about the biceps is it spans two different joints. It spans both the shoulder joint, look here, it's attaching here to the scapula, and it's also spanning the elbow joint. Okay, so it crosses two different joints, and so therefore it can affect or bring about two different movements. Because it's spanning two different joints, it can bring about flexion of the arm at the shoulder, but it can also bring about flexion of the forearm on the elbow. Because it's crossing two different joints, it can bring about angular movement in two different joints. Now, is the attachment direct or indirect? It's indirect. You see tendons there. Now, from a microscopic standpoint, these numbers aren't important. Okay? The point here is that the fibers, the cells of skeletal muscle can vary quite a bit in both diameter and length. Some muscle cells are very short, some of them are very long. Some are thinner, some are thicker. But each is a long cylindrical cell with multiple nuclei deep to the cell membrane. Notice the cell membrane is called a sarcolemma. So, for muscle, the cell membrane has a unique name, sarcolemma. Now, the cytoplasm within muscle can be called sarcoplasm. So again, within muscle tissue, the cell membrane and the cytoplasm have unique or special names. They're specific names. So when I say sarcoplasm, I'm referring specifically to cytoplasm within muscle cells. Okay. Early in the semester, I said that we store glucose as a larger molecule called glycogen. You remember that? And we said and we said glycogen is stored in two places, liver and skeletal muscle. It's stored as granules called glycosomes. So the sarcoplasm has glycosomes, a source of energy. And it's got a unique oxygen-binding protein called myoglobin. Myoglobin is a pigmented protein, very similar to hemoglobin, a pigmented protein within red blood cells. When oxygen binds to myoglobin, it gives the muscle cell a reddish appearance. Just like when oxygen binds to the hemoglobin inside of red blood cells, that blood turns bright red. Okay? So, the myoglobin is a way that the muscle cell has to store oxygen, just as the glycosome is a way it has to store glucose. Remember that oxygen is going to assist with the breakdown of glucose to generate ATP. Now, otherwise, the cells of the fibers have the usual organelles, okay, like nuclei, rough endoplasmic reticulum. Okay. But in the case of skeletal muscle, we're going to call the endoplasmic reticulum sarcoplasmic reticulum. Again, this root sarco refers to muscle. So sarcoplasmic reticulum would be the endoplasmic reticulum in muscle tissue. Now, a unique component of muscle cells is myofibrils and T-tubules. I'll talk about myofibrils briefly right now if I can find my model. Muscle cell. You all see a model of a skeletal muscle cell anywhere here? You think you see it? I'm going to go look for it because I think I can find
Is
it.
it? Go ahead.
No. I'm going to find it.
Oh.
Do not do that. Pour me some. Do not do that. You know how I feel about that type of stuff. But I had like 5 you stingy person.
What was the on the eye? You know, like it has a ciliary body and then like something's erotic. Or
Okay. This is a model of a skeletal muscle cell. Now, this is the cell membrane here. What do we call a cell membrane?
the
The cell membrane of a muscle cell.
The
The
Sarcolemma.
sarcolemma. Now, this is the connective tissue sheath. What is that called?
The outside.
The,
The muscle. Is that a muscle fiber?
The endomysium.
endomysium.
So, this is the cell membrane, the sarcolemma. This is the endomysium. Okay, now, if we look at the cell in cross-section, remember the nuclei are peripherally located. So that's the nucleus here. That's one of the nuclei. And it's just deep to the sarcolemma. All this pink stuff here is sarcoplasm. That's the cytoplasm. But you see abundant myofibrils, these rod-like elements within the cell. These are called myofibrils. That's unique to muscle cells. Other cells do not have myofibrils. And see anything else here? Well, what's that?
Does that have bands like the band-aid?
Well, this is just a single cell. If I were to show you an entire muscle, an organ, you know, of course, if this is the size of the cell, the organ would be much bigger. We kind of give you the diameter of this room. Then you would see veins. But we're just demonstrating.
No, like the bands. Like the
Oh,
band-aid.
bands, yes, yes. If you look at the myofibrils, they're alternating dark and light bands. Okay, and they're in register, meaning that the dark band of one myofibril is lined up with the dark bands of the ones around it. So the dark and light bands are in register, which gives the entire cell, and in turn the entire muscle, a banded appearance, a striated appearance. Of course, this is an axon terminal that's providing innervation to this particular cell. So we said these rod-like structures are called myofibrils. Okay? They're densely packed, rod-like contractile elements, and they contain the muscle proteins that are responsible for contraction. These muscle proteins have names: myosin, actin, troponin, and tropomyosin. So you're going to commit these names to memory. You're going to be using these terms all the time. You're on fleek today. Okay? Welcome. So myosin and actin are structural proteins that interact to bring about muscle contraction. Whereas troponin and tropomyosin are considered regulatory proteins. Okay? They're not structural, but they help that contraction to occur. And these myofibrils, as you can see here, make up most of the volume of the cell. Okay? And we said that their arrangement is such that that you see a perfectly aligned pattern of dark and light bands. Now, the dark bands, we're going to call these A bands, and the light bands, let's call those I bands. So dark bands are going to alternate with light bands. Dark bands are A bands, and light bands are I bands. You remember it like this, the second letter. The second letter in dark is A, the second letter in light is I. Yeah. Okay, got it covered. So we're looking here at this muscle cell, and we're seeing dark band and light band and dark band and light band and dark band and light band. The dark bands are the A bands, the light bands are the I bands. And again, remember that all these myofibrils are in register, so they line up. What's nice about this picture is you see some mitochondria. Remember, mitochondria are the powerhouses of the cell. They're used to produce energy. And of course, muscle cells require a lot of energy. Now, each of these myofibrils these myofibrils are these long rods. They are made up of smaller units called sarcomeres. Okay. A sarcomere is a contractile unit that extends from one Z disc to the next Z disc. And I'm drawing in Z disc here. Okay. So the region of a myofibril between two successive Z discs, and these Z discs are made up of a type of protein. Okay. That region between two successive Z discs is called a sarcomere. So along the myofibril, you have multiple sarcomeres. Okay.
Sarcomeres are the singular form.
A sarcomere is singular, sarcomeres is plural. Okay? And these sarcomeres are made up of myofilaments. These myofilaments are either thick filaments or thin filaments. Okay? Thick filaments extend the entire length of an A band, whereas thin filaments extend only across the I band and part way into the A band. So at this point, what I'm going to do is to draw a sarcomere. And did you bring your colors? Because if you bring your colors, it makes this part easier. This is something you'll have to do on the test, is to be able to draw a sarcomere. And if you don't have colors, it's okay, but it's not going to be quite as nice.
I'll just record you.
Okay. And the exercise of drawing these sarcomeres is going to help you when it comes to the test, because you're developing memory, muscle memory. It's going to help when you repeat it, when you keep drawing sarcomeres, then it commits you to memory. So remember, sarcomeres are filled with thick and thin filaments. So the thick filaments are going to be the dark ones I'm drawing here. But we said that sarcomeres are not only going to contain thick myofilaments, but also thin ones. So I'm going to draw some thin myofilaments, which will be color-coded pink. So again, thin myofilament, and over here you have thick myofilament. Now, the thick myofilaments are going to be tethered together by a band of proteins called desmin. And this protein forms what's called an M line. So an M line is going to sit right in the center of this column of thick filaments, and it's going to connect the thick filaments together. Now, in the same way, you're going to have a band of proteins that tether together the thin filaments. And this band of proteins that tethers together the thin filaments is referred to as the Z-disc. I'll keep it down just a little bit. All right. So now, the A band is defined as this region between the beginning and the end of the thick filaments. This makes up the A band.
Just the middle part? Or the whole
Right, no, the part from beginning to end of the thick filaments, this is the A band. Okay? Now, let's draw in this entire region between one Z-disc and the next Z-disc is referred to as the sarcomere. Sarcomere. Okay. Now, I'm going to draw in, for the purpose of clarity, the next band of thick filaments. Now, this zone here that's composed only of thin filaments is called the I band. So this zone here, composed only of thin filaments, is the I band. Okay. So you can see how this myofibril containing multiple sarcomeres. This is one sarcomere, but here's going to be another sarcomere here. There's going to be another sarcomere here. It represents a repeating series of I bands or light bands and dark bands. I bands and A bands. The A bands, though, include only this area between the beginning and the end of the thick filaments. Notice that the A band is going to include some overlapping thin filaments, okay? But it's by definition, it's the region between the beginning and the end of the thick filaments. The I band is that region which only contains thin filaments. And again, extending down the center of the I band is a Z-disc. This region between two Z-discs is called the sarcomere. Now, you're going to notice that within the dark band of the A band, there's a region which consists only of thick filaments. There are no overlapping thin filaments in this central region. That would be called the H zone. So the H zone is the central portion of the dark band or the A band that contain only thick filaments, no overlapping thin filaments. And at the center of the H zone, of course, is your M line. So having drawn this once, it's going to make it easier to draw it again. But the key is repetition. On the test, you will be asked to draw a sarcomere. Okay? That's going to be crucial, that you practice doing this. So thin filaments do not overlap thick filaments in the lighter H zone. This M line contains a protein called desmin that tethers together these thick filaments. Okay, now I'm going to give you exactly 5 minutes to draw what I'm drawing here, a sarcomere. Draw it and label it and make it accurate. The person who does the best job gets extra credit. Study of 13th. Did he spill a couple of these? Did you eat them all?
Not yet.
I'm feeling hypoglycemia.
Okay.
You understand that? You know, this is not quite accurate. If this is the thin filament, it wouldn't sit in the A band. Like, in other words, you describe an A band here is being made up predominantly of thin filament,
Oh.
but it's not.
Okay, it's the thing.
That's messed up.
Yeah.
Let's look at this one. Okay, not bad. Go ahead and finish up my own. Label.
Do you still want the C disc on these?
But everything. Remember, the A band's gonna extend all the way to the end. Kind of cut it short a little bit. And you're overlapping your I band here. The I band and the A band do not overlap. The I band is the part that only contains thin filament. So,
This thing's going out.
if the I band's extended from here to here, it would include any part of that A band.
Oh, see, I was just saying blue. Just with blue. That's why I put.
Right, but it doesn't include all the blue.
Okay.
It only could
Includes the blue in here.
the blue that's not overlapping
Yeah.
the thick filaments. So
Okay.
make sure so that's not quite correct.
Okay.
So let me take the three that are structurally accurate. You finished?
I was just labeling.
Go ahead, finish label.
I just twisted.
Right, yeah. So these two on the ends are not quite accurate. So we have to exclude their participation in this contest. But it's good because you guys have a lot of points already.
Okay.
Okay. You done?
I just added the thin film.
Yeah. I like the way you color coded it. That's very nice. Now let's take yours. Now, here's the deal. You cannot vote for your own, and I will be the tiebreaker if necessary. So I want everybody to come look at these, and this is going to be number 1, this is going to be number 2, and this one is going to be number 3. Okay?
Wait,
Now,
you said you can't vote for your
you
own?
cannot vote for your own.
What?
No, no.
So if you think your own's the best, then you have to vote for the second best one. Now, what I'm going to do is I'm going to create a little piece of paper here, and I want you to come tell me 1, 2, or 3. Okay. All right, Alex. Okay. Okay. Is that your own?
I mean 3, sorry.
3. Okay. All right, I think we have a winner. The winner is number 2. Who did number 2? Okay, so Hunter is going to get 10 extra points. All right, so now what I want you to do is and see if it conforms to all these definitions. A sarcomere is a region between two successive Z discs. Your drawing should demonstrate that. That the sarcomere is composed of thick and thin filaments. Thick filaments extend across the entire length of the A band. So the A band consists of the beginning to the end of the thick filaments, whereas thin filaments extend across the I band and partway into the A band. Okay. Now, that part of the thin filaments that does not extend or doesn't overlap the thick filaments, that's the I band. The Z disc, those are the proteins that anchor the thin filaments and connect myofibrils one to another. And finally, thin filaments do not overlap thick filaments in the lighter H zone. The H zone is the center of the dark band where there are no overlapping thin filaments. And that M line is composed in part of desmin, which connects the thin filaments. So this is a little bit more sophisticated drawing here. This is a little bit more sophisticated drawing where you can see the dark bands here, and this area in between, I band, another dark band. Again, the region from Z disc to Z disc would make up the sarcomere. Now let's suppose we took this myofibril and put it under a microscope so we can magnify it. Then it looks a little bit different. Now we can see that the thick filaments here, for instance, are made up of individual molecules. The thin filaments, which make up the I band, a different type of molecule. Okay? So thin filaments are composed of the protein myosin. Okay? Each myosin molecule, it says, has a rod-like tail and two globular heads. So it's going to look like this. Like a golf club that has two different heads. So this is the rod-like tail of the myosin molecule. These are the two globular heads. Okay. Whereas the thin filaments are composed of the protein actin. And these actin subunits or molecules form a helical polymer. Okay, so let's look at this. Individual subunits are going to become G-actin. And that's what I'm drawing here. That's one component of the helix. Here's the other component. Okay, so we have kind of a helical configuration to the thin filaments, and each of these subunits is made up of G-actin. The entire helical molecule would be referred to as actin. Okay. Now, each of these G-actin subunits has a binding site in the center. This is called an active binding site. Right? So, this is what actin looks like. Now, myosin looks like this. These globular heads of the myosin molecules are going to be attracted to these binding sites, these active binding sites on actin. Okay, so during muscle contraction, they're going to link up. They're going to form what's called a cross bridge. Tropomyosin and troponin are regulatory proteins that are going to coordinate this contraction. Right? Now remember, this is what myosin looks like. But to form a thick filament, you have to have a bunch of these myosin molecules bundled together. And notice that when you bundle these myosin molecules together to make a thick filament, that the globular heads project out of the thick filament at all angles. Okay. Here's the actin molecule, which makes up the thin filament. Each one of these little subunits is a G-actin subunit. And And notice this little dark area. That's the active binding sites to which the myosin heads are going to be attracted. They're going to bind. But they only bind if this binding site is uncovered. And what you see at rest here is this long filamentous protein called tropomyosin. Tropomyosin at rest is covering the binding sites. It's held in place by another regulatory protein called troponin. You see these troponin molecules? They look like little snowmen here with three little balls. Three balls. Okay. Now, one of those little balls is going to be attached to troponin. Another component is attached to tropomyosin. The third ball that's in the center of the molecule, the third component, is going to have a binding site for calcium. So again, troponin has three components, one of which binds to actin, another binds to tropomyosin, holding it in place so that it covers the binding sites, and then the middle component is going to be a site where calcium can attach. Okay. So, in a muscle, okay, we're displaying now part of a thick filament. Remember, a thick filament extends from the part of an A-band. An A-band extends from the beginning to the end of the thick filament. There's some overlap here with the thin filaments made up of actin. The central region here would be called what zone?
H.
That's the H zone where there's no overlap. Okay? But what's going to happen during muscle contraction is these myosin heads are going to attach to binding sites along the actin molecule. And then when they attach, the myosin heads pivot like this. And then they're going to detach. And they're going to attach again to a different binding site and pivot. So that we're going to have a series of ratcheting movements whereby the thick and the thin filaments overlap to a greater extent. And as they overlap to a greater extent, the muscle contracts. The sarcomeres get shorter. Does that make sense? This is called the sliding filament theory of muscle contraction. Very important. Okay? Now, before we can go there, though, we have to talk about some other things. Yeah. Remember we talked about the special name for the endoplasmic reticulum? In muscle, it's called a sarcoplasmic reticulum. The sarcoplasmic reticulum surrounds each myofibril. So it runs longitudinally surrounding each myofibril. And if you think of my forearm here as being a myofibril, the sarcoplasmic reticulum is going to surround it like a crocheted sweater. You ever see a crocheted sweater? Okay. And at certain intervals, okay, so here's the myofibril here, okay, and here's the crocheted sweater in blue. That's the endoplasmic reticulum, or the sarcoplasmic reticulum. Notice that at certain intervals, and the intervals are between, at the junction of the I band and the A band here, you have cross channels within the sarcoplasmic reticulum. Okay? These cross channels that run perpendicular to the direction of the myofibrils, okay, these are called terminal cisternae. Terminal cisternae. Okay? They're part of the sarcoplasmic reticulum. And at every AI junction, you see these cross channels, these terminal cisternae. Now, running between a pair of these terminal cisternae is this yellow structure. This yellow structure is an invagination of the sarcolemma. In other words, part of the cell membrane extends inward and travels between the terminal cisternae surrounding each myofibril. This triad, consisting of a pair of terminal cisternae and the intervening T-tubule, this is referred to as a triad. Now, the idea is that in muscle, the sarcolemma carries the action potential. In other words, the action potential travels along the sarcolemma, and then where the sarcolemma dives deep into the cell, forming the T-tubule, that action potential then travels into the cell between the terminal cisternae. So the T-tubule is a way in which we can carry the action potential from the surface of the cell deep to the interior. Now, remember the T-tubule has a close association with the sarcoplasmic reticulum. It's located between a pair of terminal cisternae. The sarcoplasmic reticulum, including the terminal cisternae in muscle cells, house calcium. Calcium is housed within the sarcoplasmic reticulum. What we're going to see later is that when an action potential travels down a T-tubule and reaches the triad, the action potential is going to trigger release of calcium from the sarcoplasmic reticulum into the sarcoplasm. Okay. So it's going to release free calcium into the cytoplasm of the muscle cell. That's what's going to happen. This is exciting. Okay. So again, let's go back. Sarcoplasmic reticulum is an elaborate, smooth metaplasmic reticulum that runs mostly longitudinally, surrounding each myofibril. Paired terminal cisternae form cross channels, and these function in the regulation of intracellular calcium levels. Elongated tubes called T-tubules penetrate the cell's interior at each AI junction. And when the T-tubules associate with the paired terminal cisternae, we refer to that group, that unit of three, as a triad. So we're getting closer to the mechanism of muscle contraction now. So T-tubules are continuous with the sarcolemma. They're just an invagination, an inward extension of the sarcolemma. So they can conduct impulses, action potentials, to the deepest regions of the muscle. These impulses, when they reach the triad, signal for the release of calcium from the adjacent terminal cisternae. So calcium is released in response to this nerve signal. Okay. So it says T-tubules in the sarcoplasmic reticulum provide tightly linked signals for muscle contraction. That release of calcium is going to bring about muscle contraction. Now, before we go forward, let me ask you to see how much you remember. That calcium, when it's freed into the cytoplasm, what we call the sarcoplasm, what protein is that calcium going to bind to? Not myosin. No. Troponin. Let's go back. Remember we said troponin looks like a little snowman with three little balls. Okay? And one of these components is attached to actin. One is holding tropomyosin in place. And the central component here is where calcium, once it's released, can bind. Now, I'm going to give you a heads up on what's the story here. When calcium is released into the sarcoplasm and it binds to troponin, the troponin molecule changes shape. It does this. Okay? In other words, it's going to displace the tropomyosin from covering these binding sites. So now those binding sites on the actin are going to be exposed, which means now that the myosin heads can attach. They can form a cross bridge. So let's see if we can understand this now. This is the sliding filament theory. Thin filaments slide past thick ones so that the actin and myosin filaments overlap to a greater degree. So in other words, during muscle contraction, if this is the thin and this is the thick, well, let's make this the thick and this is the thin. Doesn't matter. They overlap slightly at rest, but during muscle contraction, they do this. Okay? And of course, the sarcomere is shortened. So does the entire muscle. So in the relaxed state, they overlap only a little bit, but upon stimulation, the myosin heads bind to the actin and the sliding begins. So what's going to happen is once you expose, this is the actin, once you expose those binding sites because you're displacing tropomyosin, then the myosin heads do this. They have an affinity, a proclivity, okay, for that binding site. And once they do that, they carry out what's called a power stroke. They do this, which then moves, which causes these two filaments to overlap to a greater extent. And then what happens is that the myosin head attaches again, and again carries out another power stroke. And so you have a series of ratcheting movements as long as those actin binding sites are exposed. Okay? So it says, upon stimulation, myosin heads bind to actin and the sliding begins. Each myosin head binds and detaches several times during contraction, acting like a ratchet to generate tension and propel the thin filaments to the center of the sarcomere. As this event occurs throughout the sarcomeres, the muscle shortens. Okay? So let's take a look now very closely. This slide here, if you can understand this slide, this is the entire basis for the sliding filament theory. You understand everything if you understand this slide. Okay? Here we see the thick filament, here's the thin filament. Here's the troponin holding tropomyosin in place. So that because the binding sites on actin are covered, the myosin heads can't attach. The muscles are relaxed. Now, the myosin heads have a site to which ATP can attach. And when ATP attaches to the myosin head, okay, the ATP is hydrolyzed. That means you break one of those high energy bonds, okay? Remember, ATP consists of adenosine and 3 phosphate groups. Okay, that's adenosine triphosphate. When this molecule attaches to the myosin head, you begin to cleave this bond here and release energy. We call it hydrolysis. When we use water to break that bond, that's called hydrolysis of ATP. And the energy released is going to be used to do this. Watch this. This is the troponin head. It's used to cock the head like this. Put it in the cock position. Okay? Once it's in the cock position, okay, like this, then if the binding sites are exposed, there's going to be an affinity such that the two attach. Now, why are the binding sites exposed here and not here? It's because calcium's released. When calcium's released as a result of an action potential traveling down the T-tubule, that calcium is going to bind to troponin, it's going to change its shape, and displace tropomyosin. Now, the myosin head, which is cocked, is going to attach to the binding site. Once it attaches, it's going to then ratchet. It's going to perform what's called a power stroke. It's going to go from this position to this position. And in doing so, it's going to slide one filament past the other. Now, as it performs the power stroke, the two ADP and phosphate groups, the two products of hydrolysis, ADP and phosphate, release. Before it can detach, another ATP molecule has to come along. If another ATP comes along and binds to the myosin head, it will detach. It not only detaches, but it's going to use the energy in ATP to then cock again. And it can repeat this series of activities. In other words, it's going to cock, it's going to attach, it's going to perform the power stroke, and then it's going to detach again. A series of movements that brings about sliding of the filaments and muscle contraction. Does that make sense? Okay. So in the relaxed position, the thick and the thin filaments overlap only slightly. But with release of calcium and formation of these cross bridges is where the myosin head attaches to the active binding sites, then we begin to see sliding of the filaments, such that at the end of contraction, look what happens. Where is our H zone now? It disappears. It gets smaller, and if you have enough contraction, the H zone disappears. What happens to the size of the sarcomere? It gets smaller. what would happen to the eye zone here, consisting only of thin filaments? It gets smaller. The only zone that doesn't contract or get smaller is the A band, because the A band is defined as the length of the thick filaments. And the length of the thick filaments doesn't change, only its degree of overlap. That's an important concept, is that the length of the thick filament does not change. Now, I'm going to tell you a story that we can act out. It will help you to remember the sliding filament theory. It's based on German folklore. You know, there are lots of stories in history of star-crossed lovers. You know, Romeo and Juliet, Samson and Delilah. You know, you've heard these stories. And these stories, build up such a following that throughout history everybody is familiar with them. Well, in German folklore, it's the same story told across generations, told across ethnicities and cultures. There are two families, just like Romeo and Juliet. I don't know, I forget what the name of the families were. What were the families in Romeo and Juliet? You all know? Capulet, right? And what's the other one? It began with an F. Montague. Okay. In German folklore, it was Acton and Meissen. Was the family names. It was based on the real term, Acton and Meissen. They had an affinity for these two lovers were drawn to each other, such that nothing could keep them apart, not even death. Okay, that's the story. But the problem is like Romeo and Juliet, Samson and Delilah, they came from different sides of the tracks, the railroad tracks. One was considered themselves a good family, the other one not so good. And so the parents wanted to keep them apart, right? And so in this little human dramatization, we need somebody to play the role of Acton, the thin filament.
Right now?
Yeah, we're going to do it. Okay, so you're going to play the role of Acton, the thin filament. And so we need somebody to play the role of Myosin, your lover. Who would you pick? Brittany, all right. So Brittany, you're going to play the role of Myosin. Okay? Now, if there were nothing that was coming between you, you two would just bind to her binding site. You would just embrace, okay, if you will. But there are parents that are coming between you, okay? And so I'm going to have Alex play the role of Troponin. I want you to think of Troponin as your mother, okay? And Troponin is going to stand between. Ooh, you shot me. You're going to stand between Myosin and Acton, okay? Okay. Now, so you're going to prevent them from away from binding. And how do you do it? Well, you hold the dad in place, tropomyosin. Okay. So, who wants to play the role of the dad?
Cancer.
All right, Kat, come over here. Do that. So what's going to happen is, this is Troponin. She's got one hand on, you're going to hold on to Acton, and on the other hand, you're holding on to the dad, tropomyosin, to make sure the dad covers her binding site. Okay? Now, here's Myosin. Myosin would like to hook up, form a cross bridge with Acton, but what's standing in the way? Tropomyosin. Okay? Tropomyosin is covering Acton's binding site, so you two can't. Now, you see each other from a distance, you're flirting, you're saying hi, but you can't link up, right? Because the mother is making sure that the dad is standing right in the way. But you're smart. You've arranged for some friends. Right. So the friends are calcium. You're going to arrange for friends to come, and so you're the friend. You're going to be a calcium. Come over here. So y'all are at this big old dance. Remember, like Romeo and Juliet, there was a big old scene where there's dancing and partying. So you're sneaking in now, okay? And you see Acton, but you see that the parents are hovering right next door and covering her binding sites, okay? Then this is where the friend comes along, okay? You're going to come along, you're calcium, and you're going to say, hi, Miss Acton, okay? So you're going to come along to kind of grab the mom, and in doing so, what happens? She pulls tropomyosin, and now y'all can embrace. Yes. Does that make sense?
Yes.
Okay? So that's what happens with muscle contraction. As long as tropomyosin is standing in the place, is standing in the way, you two can't form a cross bridge. Once calcium is released, binds to troponin, it changes the shape of troponin such that it pulls tropomyosin off that binding site. And now you can embrace, you can form a cross bridge in muscle terms. Okay. So you would tropomyosin, I think of you as the dad. You were troponin, the mom. You were the thin filament, which is actin. You were the thick filament, which is myosin. And you were a calcium ion. Does that make sense to everyone?
Yeah.
Okay? And when you think of it that way, it's kind of a lovely story. Isn't it?
Exactly.
Right. And so, and again, remember that it's not, so when you form a cross bridge, it's the series of movements. Okay? Myosin is ready. He's cocked. Okay? The ATP has caused cocking of the head. So once that binding site is exposed, fine. Bam. You know? And then power stroke. ATP attaches, relax, cocks, binds, power strokes. Okay? So it's a series of ratcheting movements through which the muscle contracts. It's kind of a heartwarming story. Okay. So, in order to contract, a skeletal muscle must first be stimulated by nerve ending. And then it's going to propagate an electrical current or action potential along its sarcolemma. Okay? Remember that that action potential is going to travel down the T-tubule and result in the release of calcium. Okay? And that calcium is necessary to trigger contraction because it binds to troponin and exposes the binding sites. This linking of the electrical signal to the mechanical event contraction is called excitation, contraction, coupling. This is how we link. This refers to the mechanism by which we link an electrical event, the action potential, to the contraction of the muscle, a mechanical event. All right, let's do this. I am going to stop here. I think that's a lot to digest. Let's see how far we went. So we covered 29. And let's see how many this has.